nuclear DNA does not undergo recombination process. Means the recombination is important. In the paternally both are nuclear, but here only maternally. That the mitochondria are often refers to as a powerhouse. You know that last. Then mitochondrial DNA encode for proteins involves in the electron transport system, which final stage of aerobic respiration, glycolysis, Krebs cycle, electron transport system. Three systems major for release of ATPs, also called. Then women mitochondrial DNA contains approximately 16.6516 in base pairs. Base pairs. Mutation in mitochondrial DNA can lead to the mitochondrial diseases. The region of mitochondria that is highly variable and non-coding is called the D loop. The base of figures, the figure-based D loop reasons are mitochondrial DNA replicates independent of the cell. Independent means self-replicated. Any self-replicated parts it can poop and it can process to the replicate in the wound. That independent. Mitochondrial DNA is located in the matrix of the mitochondria. That is simple. In inertancy pattern of mitochondrial DNA is maternal. The mitochondrial DNA codes for 22 tRNAs and 2 RNAs in human beings. Then mitochondrial DNA is more prone to mutation than nuclear DNA because it lacks dash mechanism, repair, DNA repair. The nucleus only DNA repairs, but here is a not DNA repair. That the mitochondrial DNA analysis is often used in forensic science testing and ancestry tracing. Identification of the DNA and DNA blueprint by forensic science. The forensic testing, which is a important identification of DNA molecular structure. The DNA molecular structure base the metabolic activities of the photosynthesis in plants. That different and how much photosynthesis process rate. The rate is depends to the Rubisco. The Rubisco activity depends on the forensic science test. That is the agrogel electrophoresis. Each mitochondria contains a dash copies of empty multiple copies. The absence of the stones in empty DNA makes it more vulnerable to the damage. Then disease like mills and LHONR caused by mutation in mitochondrial DNA. Diabetes disease, any parental. So upcoming to the parental disease, all is a response to the mitochondria. That is a mitochondria. That process, that location of genes located on the mitochondrial DNA. That mitochondrial DNA is responsible to the then parental disease carries to the daughter generation. That the mtDNA was first sequenced in 1981 year. Mitochondrial DNA is transmitted through the maternal line. That's maternal line. The term mitochondria Eve refers to the most recent common female and ancestor of the women through the maternal line. Female is also major role to the carries of inheritance characters of the DNA from the parent to the daughter generation. That the chloroplast DNA. So what is the chloroplast DNA is generally dash in shape, circular dash, simple type. The chloroplast DNA found in the dash